With over a year and a half of daily discussions, news feeds, and dire warnings regarding the ongoing pandemic from various experts and laypersons alike, I'm sure I speak for the majority when I say the presented information is often confusing, contradictory, and completely overwhelming. In today's video, we're going to talk once again about SARS-CoV-2 and COVID-19, but for clarity, I want to break it down into three discrete segments. First, presenting the contemporary facts as we currently understand them. In this section, we will demonstrate how our cells replicate and use their genetic material to produce organ-specific structural and functional proteins, how viruses infect and subsequently commandeer that protein-making machinery to replicate themselves, and how the current vaccines help mitigate the clinical impact of a SARS-CoV-2 infection in susceptible individuals. We will then use those facts to speculate on the potential evolution of the virus and the proposed mechanism of action of various treatment options available. The final segment will be dedicated to my own personal opinions on the topic, but will be clearly presented as such so as not to misconstrue any of that as expert commentary, an issue I personally have with the majority of articles presented in the lay press today. So let's get started with the facts. In the human body, a parent cell divides into two daughter cells, splitting the cytoplasm and nuclear material equally between the daughters. The critical genetic material, or template, for each and every one of us is encoded in the double-stranded DNA within the nucleus of the cell. During division, this DNA needs to be perfectly replicated, with each daughter cell receiving an identical copy to carry out its assigned function. This is done through the process of mitosis, where the helical strand is unwound and each strand duplicated by pairing each of the four nucleotides labeled T, A, C, and G with its complementary base linking T to A to T, C to G, and G to C. With each daughter now complete with its full complement of DNA, it needs to start producing structural and enzymatic proteins to carry out its assigned duties and contribute to the functional activity of that particular organ or part of the body. This is done through ribosomes, an organelle in the cell cytoplasm that can read genetic material and translate that code into the necessary proteins that keep us all alive and functional. All ribosomes consist of two subunits, one larger than the other. Humans have 80S ribosomes consisting of a 40S and a larger 60S subunit. Prokaryotes, or single-celled organisms like bacteria, have 70S ribosomes consisting of 30S and a larger 50S subunit. This distinction between the eukaryotic and prokaryotic ribosomes may have relevance when we're talking about potential antibiotic therapies for SARS-CoV-2 in a moment. Our DNA never leaves the protection of the nucleus, but the information it contains needs to get into the cytoplasm of the cell where the ribosomes reside for translation into proteins. This is accomplished through RNA polymerase. In a process called transcription, RNA polymerase reads the coding strand of DNA and produces a complementary single-stranded messenger RNA, or mRNA, again using complementary base pairing. As before, C always pairs with G, G with C, and T with A in the mRNA strand. However, the A base in the DNA sequence is transcribed into uracil, or U, in the mRNA strand. This transcribed messenger RNA then leaves the nucleus and enters the cytoplasm. The strand of messenger RNA encodes for 20 different amino acids, the basic building blocks of proteins. Specifically, every 3 bases encodes for one of the 20 amino acids. Hence, this grouping of 3 nucleotides is called a codon. The resulting linear strand of amino acids, or polypeptide chain, then fold into a complex 3-dimensional structural or functional protein. Viruses come in various configurations, but all have basically the same functional components. Regardless of the subtype, all viruses are essentially a strand of genetic material encapsulated by structural and functional proteins that help protect and transport their genetic material to a susceptible host. Another distinguishing feature is the fact that viruses do not have any machinery to reproduce or transcribe their genetic material into new viral particles and therefore are dependent on the host organism for those functions. Viruses can contain various forms of genetic material, including single and double-stranded DNA, as well as single and double-stranded RNA. Because of these genetic variations, viruses will interact differently with their hosts. SARS-CoV-2, the infecting agent for COVID-19, is a single-stranded RNA virus also known as messenger RNA or positive sense RNA. There are 4 antigenic proteins present on the surface of the SARS-CoV-2, all designed to safely transport and deliver the genetic material for replication and new viral production. The multiple spike proteins on the surface of the viral particle seek out a particular surface protein on the host cells. In this case, the transmural zinc-containing metalloprotein called ACE2. The ACE2 surface protein is ubiquitous in the body, present on the surface of innumerable cells comprising multiple organs, including the lungs, blood vessels, heart, GI tract, olfactory and sheathing or glial cells, and kidneys, which may explain some of the clinical features of SARS-CoV-2 infections, including pneumonia, blood clots, myocarditis, GI distress, anosmia or loss of smell, and renal failure. The exact mechanism of action between the cells of our body and the SARS-CoV-2 viral particle is not completely understood, but for the purpose of this discussion, we'll assume the virus is incorporated into the cell cytoplasm intact through a process of endocytosis. After attaching to the ACE2 surface protein, the viral particle becomes encapsulated in a spherical portion of the host cell membrane, forming a vacuole. At this point, to initiate viral replication, the viral particle needs to deliver its genetic material to the host cytoplasm. Enter the lysosome. Lysosomes are small organelles that contain a number of chemicals like proteases used to break down the protein structures of food and pathogens, sort of the stomach for each of the body's cells. For optimal function of all these chemicals and proteases, the internal pH of the lysosome is acidic at about 4. This will be important when we're talking about potential treatments in a moment. The lysosome and endosome fuse, allowing the contents of the lysosome to work on the viral envelope and surface proteins. Again, this process allows the envelope and endosome membrane to fuse, providing a conduit for the viral genome to enter the host cell cytoplasm. Coronavirus genetic material is a positive sense RNA genome of about 30,000 bases, quite large considering some viruses contain a few dozen bases. Positive sense means that the viral RNA structurally resembles messenger RNA or mRNA of our host cells and therefore can be read and translated directly by the host ribosome. So the single strand of RNA from the virus can be read directly by our ribosome to make proteins, but there is no way to use the host cell organelles to actually replicate the genetic code to make more viral particles or reproduce. Remember, all our DNA duplication takes place in the confines of the nucleus, isolated from the cytoplasm. Fortunately for the virus, and unfortunately for us, the virus has figured this out. The first 20,000 bases, 2/3 of the entire genetic code, are used in the production of a complex protein molecule called RNA-dependent RNA polymerase, or RNA replicase. This protein molecule serves multiple functions in the viral replication process and is important as a possible target for therapy. The RNA-dependent RNA polymerase can now read the viral RNA and make a complementary RNA strand linking adenosine to uracil and guanine to cytosine. The new RNA strand is basically a matching template of the original positive sense RNA and is therefore called a negative sense RNA strand. This strand cannot be read by our ribosomes to make protein, but can be used by the newly assembled RNA polymerase to make new positive sense copies of the genetic code, replicating the original RNA. The RNA transcriptase can read the negative sense RNA template a second time and produce small pieces of positive sense genetic material that encode for the 4 structural proteins of the new virus. These smaller pieces are called subgenomic since they only include small portions of the entire genetic code. The smaller RNA fragments are then read by the host ribosome to produce the structural parts of the new virus. The new structural and surface proteins are then assembled into new viral particles, loaded with a copy of the genomic RNA, encapsulated in a membrane within the cytoplasm, and delivered outside of the cell through excitosis or budding. Once outside the cell, the process begins again. When our bodies are exposed to a pathogen Our immune systems, predominantly through various members of the white blood cell or leukocyte line, defend us against that invasion by recognizing the virus or bacterium as foreign and mounting a coordinated attack against the same. The major players in this process are the B cells, T cells, and the mononuclear phagocytic cell line, which include monocytes, macrophages, and dendritic cells. For our discussion, we will be focusing on the dendritic cells, also known as antigen-presenting cells or APCs. Looking back at our coronavirus, there are 4 major structural proteins on the surface of the virus that can serve as potential identifiable antigens for our immune systems, including the spike or S protein, nucleocapsid or N protein, membrane or M protein, and envelope or E protein, all designed to house and protect the delicate genetic material in its hollow spherical core. The dendritic cells phagocytize and digest or break up the virus or bacterium. Once disassembled, these cells can then present some of the characteristic protein structures or antigens of the invader on their cell surface. The dendritic or APC cell then travels to the lymph nodes where it presents its findings to other members of the white blood cell line. The T cells learn the various antigens and use them as targets to identify infected host cells in the body. They then alert the immune system to the invasion, recruiting other members of the leukocyte line to assist in the battle and hopefully destroy or disable the virus before it can gain access to a host cell. The T cells can then destroy the already infected host cells, preventing them from replicating new viral particles. The B cells analyze the various viral antigens and develop antibodies that are specifically designed to attach to the freely circulating viral particles. One B cell line may produce antibodies to the spike protein, another to the envelope, and others to the nucleocapsid and membrane proteins. Once trained, each of these B cells turns into plasma cells or memory B cells. Plasma cells are basically antibody factories pumping out the newly developed viral antibodies that can then attach to the specific protein antigens of the new viral particles, either deactivating them directly or providing a target for destruction by other members of the immune system. Each B cell or plasma cell produces a single species of antibody designed to attach to its specific protein antigen that was initially used to train the cell. Specifically, the spike protein plasma cell produces antibodies which attach only to the spike protein of the virus. The nucleocapsid plasma cell produces antibodies which only attach to the nucleocapsid protein of the virus. The envelope plasma cell to the envelope protein of the virus, and the membrane plasma cell antibodies which are specific for the membrane protein of the virus. With a new or novel pathogen, this leukocyte training process takes time, during which the virus or bacterium continues to multiply and infect other cells of the body, sickening or killing the host in the process. This is where the memory B cells come into play. Hanging out in the lymphoid organs of the body like the lymph nodes and spleen, the B cells can be quickly mobilized into antibody-producing plasma cells if a new infection from the same virus or bacterium is encountered in the future. Specifically, if the immune system encounters the identical antigen in the future, the memory B cells rapidly divide, convert to plasma cells, and begin flooding the host with antibodies to quickly fight the infection. In other words, the host has now developed an immunity to this particular virus, reducing the clinical impact of subsequent infections. The primary objective of any vaccination is to train the body's immune system to recognize an infecting particle and develop antibodies to the same before an actual infection. There are currently 2 types of vaccines against COVID-19, mRNA and DNA, both of which use a man-made strand of genetic material to encode for the spike protein of SARS-CoV-2. The mRNA vaccines include Pfizer and Moderna, while Johnson & Johnson, AstraZeneca, and Inovio are all examples of DNA vaccines. Let's start with the mRNA products. Scientists have figured out the segment of the viral genome that encodes for the spike protein of the coronavirus and are using this subgenomic genetic sequence as the core of their vaccination products. Since this sequence resembles messenger RNA, it is known as a messenger RNA or positive sense RNA vaccine. However, to do its job, the RNA sequence needs to remain intact while it is transported and incorporated into the host cell cytoplasm. Historically, this was accomplished with a viral vector. The virus is loaded with the genetic code, and the virus was then used to infect the host cell with the new genetic sequence. However, the current vaccines use a structure called a lipid nanoparticle for transport and delivery, similar in structure to the lipid transport vehicles of the body, including chylomicrons, low-density lipoproteins, or LDL, and high-density lipoproteins, or HDL. They consist of a phospholipid shell which engulfs and transports the genetic code through the bloodstream. It's common knowledge that oil and water don't mix. To transport fatty molecules through our bloodstream, the outer wall of all lipid transport vehicles contain the charged phosphate group of the phospholipids and the charged portion of the free cholesterol molecules, allowing the entire sphere to be miscible with the polar water molecule in the plasma, while the center of the sphere contains the nonpolar fatty components of the phospholipids and cholesterol, storing the nonpolar triglycerides and cholesterol esters for transport and cellular delivery around the body through the bloodstream. Similarly, the lipid nanoparticle has a phospholipid membrane that contains and protects the delicate mRNA encoding for the spike protein to the cells of the body. Once the particle enters the cytoplasm of the host cell, the mRNA is delivered to the host ribosome, where it is translated into the spike protein of the SARS-CoV-2 virus. Remember, there are no provisions to replicate messenger RNA in human cells, as the molecule is produced through RNA polymerase from a DNA template in the nucleus. As such, protein production is limited by the volume of subgenomic material contained within the vaccine volume. Once formed, the spike proteins are then released from the cell and become available to train the immune system to produce antibodies against the spike. Remember, since the genetic sequence solely encodes for the spike protein, only spike B cells, plasma cells, and antibodies will be produced with the current mRNA vaccine. Memory B cells are created, and subsequent infections with the actual SARS-CoV-2 virus activate the memory B cells, quickly ramping up antibody production to the spike protein of the actual virus. The antibody binding to the spike protein may physically block the virus from entering the host cell through the ACE2 surface protein, or could mark the virus for destruction by other members of the lymphocyte community. As the name implies, the DNA vaccines use a man-made segment of double-stranded DNA, just like our own native genetic material, to encode for the spike protein of the SARS-CoV-2 virus. Remember that our DNA is only replicated and translated into mRNA within the nucleus of our cells, and therefore the DNA vaccines need to gain access to the host cell cytoplasm, just like their mRNA counterparts, but then need to deliver their genetic payload into our cell's nucleus for replication and translation. To accomplish all of this, a different transport vector is required. Johnson & Johnson uses a simian adenovirus. Adenoviruses are one of the infecting agents responsible for the common cold in humans. Simian means monkey, and therefore, the simian adenovirus causes cold-like symptoms in monkeys, but is also an effective vector for delivering DNA genetic material in humans. The simian adenovirus is loaded with the double strand of DNA encoding the viral spike protein, is injected into the body, and the viral vector then seeks out and delivers this genetic code into the susceptible cells of our body. Once incorporated into the nucleus, the process of genetic transcription and translation begins, turning our cells into a SARS-CoV-2 spike protein factory. Based on the facts presented, we're now going to make some assumptions. The clinical response to a SARS-CoV-2 infection is quite variable, ranging anywhere from nearly asymptomatic to death, and we now know that certain comorbid factors can almost predict how a patient is going to behave. Obese diabetics with hypertension and COPD seem to be most susceptible to a severe clinical course, and we now have some theories on why that may be. Remember that SARS-CoV-2 seeks out the ACE2 surface protein as a gateway for host cell entry. People often refer to this structure as a receptor for SARS-CoV-2, but I personally feel that that label is misleading. ACE2 is actually part of the renin-angiotensin-aldosterone system, or RAAS. Involving the kidneys, liver, lungs, blood vessels, and adrenals, the renin-angiotensin system is a closed-loop feedback mechanism that helps maintain renal blood flow. The kidneys are designed to filter the nitrogen waste products of protein metabolism from our bodies. To function normally, They need a steady flow of blood, receiving about 25% of the total cardiac output. A drop in blood pressure, either systemically from heart failure or locally from a narrowing of the renal artery supplying blood to the kidney, causes the kidney to secrete a chemical called renin into the bloodstream. Renin then links to a protein hormone produced in the liver called angiotensinogen, converting it to angiotensin 1. A second enzyme produced in the lungs, called angiotensin-converting enzyme, or ACE, then links to angiotensin 1, converting it to angiotensin 2, the active configuration of the hormone. Angiotensin 2 then acts on both the peripheral arteries throughout the body and the adrenal glands. The arteries constrict, and the adrenal glands produce another hormone called aldosterone, which causes the kidneys to resorb more salt and water from the urine. The combination of vascular constriction and saltwater retention raises the systemic blood pressure and restores blood flow to the compromised kidney. Looking back at our ACE2 surface protein, the active form of ACE2 is produced by an additional enzyme called SHEDase. SHEDase cleaves the external component of the ACE2 protein and releases it into the bloodstream. The cleaved ACE2 then interacts with angiotensin 2, converting it into angiotensin 1,7. Angiotensin 1,7 is a powerful antioxidant and vasodilator, dilating the peripheral vessels of the body and eliminating the action of angiotensin 2 on the adrenals. Angiotensin 1,7 lowers blood pressure and is basically the counterbalance to the renin-angiotensin system. Hopefully from this explanation you can now appreciate that the renin-angiotensin-aldosterone system and ACE2 surface proteins are opposing biochemical processes that are in a fine balance determined by the patient's baseline health. Obesity, smoking, and a poor diet lead to heart failure and atherosclerotic narrowing of the blood vessels, both of which will invoke the renin-angiotensin-aldosterone system to maintain renal blood flow. On the other hand, regardless of age, an individual maintaining a healthy diet, regular exercise, and sleep will likely maintain adequate renal blood flow, causing SHEDase to cleave the ACE2 surface protein, reducing systemic blood pressure, and creating a generalized anti-inflammatory effect. But how does all this affect the clinical course of a SARS-CoV-2 infection? Remember that the SARS-CoV-2 virus is looking for the ACE2 surface protein as a doorway to enter our cells. If the external component is shed, the virus can no longer recognize that region as a potential entry portal to begin viral replication. In addition, the soluble form of ACE2, now shed from the cell membrane surface, can not only counteract the effects of the renin-angiotensin-aldosterone system, but can also bind to the spike proteins of the circulating viral particles, preventing them from even interacting with fixed ACE2 sites still on the cell membrane surface. In other words, your baseline health appears to be a significant, or possibly the most important factor in determining your clinical course with a SARS-CoV-2 infection. There are two more items I want to cover in the assumptions section. Number one, we're going to review some of the approved and repurposed medications used for COVID-19 and their suggested mechanism of action. And number two, the expected evolution of the SARS-CoV-2 virus. Hydroxychloroquine, doxycycline, azithromycin, and ivermectin are all existing medications that have reported anecdotal efficacy against SARS-CoV-2. Vitamin D, while not a medication per se, seems to help suppress symptoms of a SARS-CoV-2 infection and therefore warrants mention. Hydroxychloroquine, an antimalarial, anti-rheumatoid medication, appears to accumulate in lysosomes of our cells, raising the pH from the acidic 4 to a more neutral 6. Again, the proteases and other enzymes in the lysosome function optimally in the acidic environment. The elevated pH may reduce the function of these chemicals, preventing uncoating of the viral RNA and essentially entombing the particle in the endosome. The antibiotics azithromycin and doxycycline are bacteriostatic drugs that don't kill bacteria directly, but stop them from reproducing by binding to their ribosomal large and small subunits respectively. This sounds promising since we now know that coronavirus uses our ribosomes to reproduce. However, remember that humans have 80S ribosomes, not the 70S units found in prokaryotes that are targeted by these bacteriostatic medications, and therefore the exact mechanism of action against viruses in humans is questionable. Remdesivir and Merck's new oral molnupiravir are both antiviral medications that reportedly lessen the clinical impact of SARS-CoV-2 infections. Both medications interfere with viral replication by mimicking nucleotides during RNA replication. Remdesivir is an injectable prodrug that is metabolized by the body into an adenosine analog. This analog is incorporated into the mRNA copy and presumably can't be read by the RNA-dependent RNA polymerase, causing chain termination and halting further replication during transcription. Molnupiravir is a prodrug metabolized in the body to form an analog of cytosine. The artificial nucleotide is inherently unstable and can change its structure to also mimic uracil. These substitutions and transitions cause extensive errors during RNA replication, resulting in lethal mutagenesis or viral death. As with most antivirals, however, they seem to be most efficacious when administered early in the course of the disease, but even then, some of the preliminary data with remdesivir suggested very limited efficacy. Merck is currently seeking emergency use authorization approval for its oral version of this class of drug. A quick word on vitamin D. For many years, scientists have suggested that vitamin D plays an important role in supporting immune health, especially when fighting viral infections. Many patients with a severe clinical course with a SARS-CoV-2 infection are noted to have low serum vitamin D levels. In fact, anecdotal reports suggest that vitamin D levels precipitously drop during an active infection, presumably due to consumption of the vitamin D molecule while the immune system battles the virus. Many doctors now advocate vitamin D supplementation to support immune health, and oral dosing should be titrated to maintain serum 25-hydroxy-vitamin D levels between 50 and 80 nanograms per deciliter. Regarding the evolution of SARS-CoV-2, as we stated earlier, viral replication for a positive sense or mRNA virus is highly inaccurate, resulting in coding errors, most of which result in viral death. However, this can also be an evolutionary advantage, as the virus can mutate rapidly and adapt to Darwinian pressures to ensure its survival. An effective virus shouldn't kill its host, but lives within it, replicating its genetic code and propagating the species. As such, assuming normal Darwinian evolutionary forces, we would expect the virus to become more infectious but less virulent with time. Finally, I want to conclude with my own personal opinions. I know that was a lot of information, but I think the review was necessary to support my positions. Let's start with the medications. While anecdotal reports on hydroxychloroquine, doxycycline, azithromycin, and ivermectin tout amazing efficacy with these repurposed drugs, the supportive data to establish causation is lacking, and many of the trials are contradictory and obviously biased. Remember that the clinical response to a SARS-CoV-2 infection is extremely variable, and in otherwise healthy individuals usually self-limiting. As such, an amazing response could simply reflect the body's ability to effectively fight off the infection and avoid a fulminant COVID-19 clinical course. Double-blinded placebo-controlled studies tease out that information and limit anecdotal bias. While I have no problem treating symptomatic patients with any or all of these repurposed therapies, I personally would not recommend anyone prophylactically taking any of these drugs until that information is available establishing efficacy and safety. In my opinion, the choice of a genetic vaccine that only encodes for the spike protein of the virus may be problematic. At first glance, this choice may seem appropriate since that is the structure that seeks out the ACE2 doorways to gain access to the host cells. Antibodies to the spike will bind to the viral surface, preventing it from interacting with our cell membranes, thus stopping the particle from gaining access to the protein-making machinery of our cells. However, remember that the ACE2 surface proteins are ubiquitous in the body, found in the lungs, blood vessels, and kidneys, as well as the heart muscle and GI tract. When these spike proteins are released into the bloodstream, they can still seek out and bind to the ACE2 metalloprotein on our cells, but our bodies now have immunity to the spike and will try to attack it as a foreign invader. As such, all of our organs now coded in these genetically encoded spike proteins become targets for our own immune system. This may explain some of the observed side effects post-vaccination, such as GI distress and cardiomyopathy. There are also reports that these spike proteins can cross the blood-brain barrier and interact with the cells of our central nervous system, resulting in symptoms like sustained vertigo or paranoia. The fallacy that SARS-CoV-2 vaccinations are going to rid mankind of SARS-CoV-2 or that vaccine immunity is better than viral immunity is absolute nonsense. In fact, mass vaccination with the current generation of vaccines may actually be counterproductive. Remember that the current vaccines encode for a single antigenic spike protein on the viral surface, whereas natural immunity with a SARS-CoV-2 infection provides memory immunity to all four antigenic proteins. Combined with the fact that these mRNA viral particles are highly mutagenic and can quickly adapt to environmental pressures, mass administration of a vaccine that only encodes for a single protein will artificially and quickly force viral mutations to render the current vaccines worthless, negating their benefits for the most susceptible individuals who need it the most. As with any intervention or procedure in medicine, we look at the risk versus the benefits. If the risk of the intervention is greater than the benefits, it would be completely irresponsible to continue with the procedure knowing that the intervention is potentially worse than the disease itself. On the other hand, if the benefits outweigh the risk, the procedure is probably indicated. The epidemiologic data collected over the last year suggests that the vast majority of otherwise healthy individuals are going to do just fine with a SARS-CoV-2 infection, with a few days of minor cold or flu-like symptoms and possible loss or a distorted sense of smell for a few weeks. However, if you're obese and hypertensive, you're at high risk for a fulminant COVID-19 clinical course and possibly dying from a SARS-CoV-2 infection. In the young, healthy 25-year-old, the risk of the disease, in my opinion, is far less than the potential risk of injecting a man-made strand of genetic material that has absolutely no clinical data to guarantee safety over the expected 55 years of that person's remaining life. However, in the obese hypertensive, the risk of dying from SARS-CoV-2 infection is substantial, and the use of a novel genetic vaccine is probably warranted to save that individual's life. Understand that the purpose of the vaccine is not to prevent you from getting infected from SARS-CoV-2, but from dying from it. At the end of the day, vaccinated or not, the only way around this pandemic is straight through it. In my opinion, we need to stratify risk and reserve the vaccine for the most susceptible members of society. As I've said from the beginning, this is never going away. All of us eventually need exposure to the native virus to develop a full complement of B and T cell memory immunity, at which point we obtain true herd immunity and simply live with the seasonal recurrence of COVID-19. In my opinion, the most important determinant of your clinical course with a SARS-CoV-2 infection is ultimately your viral load. Because of the haphazard way these mRNA viruses replicate, once you reach a critical mass of viral particles, you overwhelm the immune system, inciting a cytokine storm and fulminant COVID-19 clinical course. One way to reach that critical mass is with a large initial viral load. If you're hanging out with a bunch of sick individuals coughing and sneezing all over you, no matter how healthy you are, you may quickly reach that critical mass of viral particles and get really sick really quick. As such, when I feel I've had a significant exposure, I try to decrease my viral load at the entry sites through the mouth and nose. I will gargle with mouthwash to attack the particles on the oropharynx and use my neti pot or nasal lavage a few times a day to reduce viral load in the nasal passage and nasopharynx. The other way to reach that critical mass of viral load is to provide more cell membrane entry portals for the virus, hence the increased risk of a serious COVID-19 clinical course in obese hypertensives. If you really want to protect yourself from infection, in my opinion, the best thing you can do, better than any repurposed medication or genetic vaccine, is get in shape. While I'm all for a body-positive attitude and feeling comfortable in your skin, at the end of the day, obesity is still a disease that eventually is going to kill you. With SARS-CoV-2, it just may be 2 or 3 decades earlier than we originally expected. Stay safe, and we'll see you next time.